Today we are discussing about the paper electrophoresis. What is the meaning of electrophoresis? Here the introduction of electrophoresis and what is electrophoresis, this will be explained in previous videos, so go and check there. Here in this class we will discuss about the paper electrophoresis. The paper electrophoresis is a separation technique. This is also one type of separation techniques where ions of different charges are separated on a medium of the paper. When we apply the voltage between two electrodes which are in contact with the paper, and the ions will be get separated by their affinity. Okay, that will be observed in this paper electrophoresis technique. And in paper electrophoresis, the primarily used to separate the mixture of amino acids are proteins and peptides. Okay, this paper electrophoresis used to separate the mixture of amino acids, proteins, peptides and other chemical substances. That is nothing but to separate the DNA, RNA particles, okay, molecules. What is the main principle of this paper electrophoresis? Here a mixture of ions that will be substances, means a mixture of ions or insoluble substances which is present in a solution. That mixture is applied on the center of the paper and then previously immersed into a buffer that is called as ionic strength. And in this paper the two trays are there which is placed across the two trays, then filled with a buffer into which two electrodes are immersed. When a voltage is applied across to these electrodes, the ions will be migrated towards to the anode and cathode based upon their charges. Okay, based upon their charges they will be separated sides. And neutral or non-ionizable substances which can be migrate to the separate sides. For example, ionic substances are moved towards to the anode and cationic substances which move towards to the cathode. Okay, ultimately there is a separation of ionic, cationic and non-ionic substances. Mainly in this paper electrophoresis the principle is separation of the ions, and the bands or spots which can get migrated by the using appropriate sprays or reagents. When we are sprayed on the chromatography paper, we can be analyze that ions. The electrophoretic mobility.